Now let's talk about the next technology, the next technique, that is AIT. AIT is artificial insemination technique. Now this is artificial insemination, so you can just make out from the name itself. The semen from the male is washed thoroughly and concentrated, and the semen, what semen contains? Sperms. The semen from the male contains sperms. It is washed thoroughly, concentrated in the lab, and then this is introduced inside the body of the female, inside the vagina or the uterus, inside the vagina or the uterus. So when this semen is injected into the female's body, then this semen, as it contains sperms, these sperms are going to come in contact with the egg. They are going to fuse. Only one sperm actually is going to fuse with the egg and fertilization will take place. So when you talk about this artificial insemination technique, then the donor male can be from outside also. So it is not necessary the donor is the husband only. The donor can be from outside also, and we have sperm banks from where we can actually have the semen that is injected inside the female bodies during this technique, that is artificial insemination technique. So let us see how actually this artificial insemination technique is there. As you all know, that is that inside the ovaries there is the maturation of the eggs that is taking place, right? So the egg is released and this egg enters the fallopian tubes and wait for the sperms to come so that fertilization can take place. But if fertilization is not taking place, means if there are the chances of infertility, then the couples can opt for these technologies that we call as what? We call as assisted reproductive technologies, as I told you earlier also. So we are discussing this technology, that is AIT, that is artificial insemination technique, and under this we have intrauterine insemination, means the semen is injected inside the uterus, right? So what is going to happen? We are going to have the semen from the male which is containing millions of sperms. So if there are 10 millions of sperms, so this is considered more favorable. Then they, under laboratory conditions, they are washed thoroughly, they are checked for their quality, quantity and other conditions, and after that through a very flexible tube, this semen is injected inside the vagina, or you can say the uterus, right? So this is how artificial insemination takes place. So the sperms are injected in such a way so that they can come as much close to the egg as it is possible. So when the sperms, they are going to come in contact with the egg or the ova, only one sperm is going to fuse, and this is going to form the zygote, and the zygote develops into an embryo into a fetus, and fetus into what? Fetus into a normal and healthy child. So this is how artificial insemination technique is there. Now the chances of success of this technique is around 20 to 40%. Right? It is around 20 to 40%. Next technique we have, next technology we have GIFT. What is GIFT? GIFT is gamete intra-fallopian transfer. It is what? It is gamete intra-fallopian transfer. And then we have ZIFT. ZIFT is zygote intra-fallopian transfer. So as the name suggests, in gamete intra-fallopian transfer, the gametes, that is the sperms from the male and the ova or the egg from the female, these gametes are transferred into the fallopian tubes. So this is gamete intra-fallopian transfer. Now this is actually in vivo fertilization. It is what? It is in vivo fertilization, means the fertilization is taking place inside the body of the organism. So it is what? It is in vivo fertilization. Fertilization taking place inside the body. Well, just remember that we have just transferred the gametes, that is the sperms from the male and the egg or the ova from the female. Only the gametes are being transferred, right? So when these gametes are being transferred, the fertilization is not taking place outside the body. like in the case of test tube baby, the fertilization is taking place inside the body of the female, that is there in the fallopian tubes where we are going to transfer the gametes. So this is how gift is there, that is gamete intrafallopian transfer. It is a form of in vivo fertilization where fertilization takes place inside the body. Then we have ZIFT. ZIFT is zygote intrafallopian transfer. Now zygote intrafallopian transfer is actually a part of, you can say, this in vitro fertilization. So as we have discussed in case of test tube babies, that fertilization takes place outside the body, and when the zygote is formed, that zygote is transferred into what? Into the fallopian tubes, where the zygote is again going to form the embryo, then fetus, and then the child. So this is how zygote intrafallopian transfer is there, means when fertilization has taken place outside in the lab condition and zygote is formed, that zygote is transferred into the fallopian tubes. And if supposingly that zygote divides, you know, more than, you can say, at more than 8 cell stage, the blastomere, 8 blastomere stage, after that it is implanted into the uterus, not into the fallopian tubes. So this is zygote intrafallopian transfer, and gift is gamete intrafallopian transfer. So you can easily make what are these technologies by the names itself, right? Gamete intrafallopian transfer, it is what? It is in vivo fertilization. And then we have ZIFT, that is zygote intrafallopian transfer. So I hope that these 4 assisted technologies, assisted reproductive technologies, they are clear to you: IVF, AIT, gift and ZIFT. Now if we talk about the success rate in case of gift, then gift is very expensive as compared to other technologies, but the success rates are comparatively higher. Success rates of gift are comparatively higher among all of these, among, you know, if you consider IVF, if you consider AIT, if you consider ZIFT, then gift has more success rate as compared to all of these, and it is more expensive as compared to others. So I hope that these 4 reproductive technologies, which are the assisted ones in case of infertility, they are clear to you. Just make out how these technologies they are used. The main application is, you know, in case of couples who cannot have a child, these technologies they help those couples to have the child, right? So that they can have a child who is normal and healthy. So these are the technologies that are nowadays used to help the couples to solve the infertility problems.